everyone, I'm Maisie. I'm one of the 10 interns, and this week we were lucky enough to talk to Dr. Roberts about his research into how chemicals can affect fish. Dr. Roberts does research into 3 areas of how these chemicals affect the fish. First off is molecular biology, which is how the chemicals affect the very cells of the fish at a molecular level. Secondly is physiology. That's how the chemicals can affect the greater structures of the fish's body. For example, like their bone structure or their circulatory system, etc. Thirdly, he studies the ecology of how the fish are affected. This means how they're being affected at a population level, or how the whole community is being affected by the presence of a chemical in the water. The first kind of contamination he studies is toxic liquid spills, for example, oil spills. These spills can create a constellation of effects. What that basically means is that the way these oil spills and chemicals affect, say, a dolphin is very similar to the way they affect a fish. What was surprising to me that he told us was that if you look at the way the oil enters a dolphin or other aquatic mammal's lungs, it actually looks very similar to a smoker's lungs. The way these chemicals build up within them over time is very similar. Now, one of the first ways he investigated the effects of oil spills on fish was through gene expression work, which is essentially looking at the fish's very DNA and seeing how it's being affected. And what he found was that the oil spills were really affecting the certain genes inside a fish's DNA that affect their eyes and how they see. Now, to figure out how the oil spills were actually affecting the fish's eyesight, he had to come up with a way to test the fish's eyesight, which is harder than you might think because you can't exactly ask a fish to read lines of letters off a piece of paper. So flicker fusion is what it's called when, for example, you're a person like me and you're looking at a ceiling fan watching it go by. And at first you can see the rungs of the ceiling fan, but then eventually it starts going so fast that you just sort of see a blur, and you'll see them flickering, which is where the term flicker fusion comes from. So basically, part of what he was testing was trying to see how long the fish can keep track of the one object before it moves fast enough that it becomes flicker fusion. So he used a species of fish that likes to follow things, and he put them inside a cylinder of water that was surrounded by a spinning cylinder with black and white stripes. Then he would twist the cylinder with the black and white stripes and watch as the fish tried to follow one specific stripe. The fish would always lock onto one. As it started spinning at a faster and faster rate, the fish would go to keep up, and they could actually swim really fast keeping up with these. But then eventually they'd reach a point where they could no longer distinguish the stripes, and they would just sit in the middle of the tank and look at the gray wall. So he used this test on fishes who had been exposed to the chemical contaminants that would be found in an oil spill. And what he found was that these fish weren't able to keep up as long or distinguish the stripes for as long. Additionally, he found that the physical eye shape of the fishes had been changed and different from those of fish who hadn't been in contaminated water. These eyes were smaller and had less clearly defined structures and layers within the eye. He even discovered that these contaminants can literally kill off the nerve cells in the eye, obviously leading to decreased eyesight in the fish. Next, he investigated the effects of mercury on both fish themselves and on an ecological level. Mercury is introduced into ecosystems both by natural causes and by humans. And in fact, 2/3 of all mercury are from anthropogenic sources, in other words, introduced by humans. The number one way us humans are putting mercury into the atmosphere and into bodies of water is through coal burning. An interesting fact about mercury pollution is that once mercury enters the atmosphere, it can remain in the atmosphere for up to 2 or 3 whole circulations of the Earth. This means that if here in Dallas we put mercury into the atmosphere, it could end up coming down in South Africa or Japan. So mercury pollution is really a global issue because no matter where you're doing it, it's going to affect the whole world. So after we pollute mercury into the atmosphere or a body of water, biological processes will convert it into organic mercury, also known as methyl mercury. One of the reasons methyl mercury is so dangerous is because it biomagnifies. What biomagnification means is that if, for example, there's a little fish in the water that's ingesting mercury from the water, that mercury will get stuck in its system. Then, if, say, a bigger fish comes along and eats 10 little fish in one day, or many more over time, suddenly that bigger fish has a much higher concentration of mercury because it's all staying in its body. None of it is leaving. Then in time, a bird will come along and eat the bigger fish, and so on and so on. So as you go higher in the food chain, these species are having more and more mercury in their bodies. This is a problem for us, because if we eat a fish that has mercury in it, all that mercury will stay in our body. So because of the threat mercury, and especially fish with mercury in them, pose to human health, all 50 states have at least one restricted body of water that is known to contain mercury, and therefore we're not allowed to fish in it or eat fish from it. Texas particularly has a very high deposition rate of mercury. So in order to investigate the mercury levels, particularly in Texas, and their effects, Dr. Roberts chose to investigate Caddo Lake, also known as the only natural freshwater lake in Texas. Furthermore, Caddo Lake actually has some of the highest concentrations of mercury, which made it a really good case study for his investigation. Dr. Roberts sampled tissues from many different organisms within Caddo Lake, and found that organisms that were higher in the food chain contained higher levels of mercury, which supports the theory I was discussing earlier of biomagnification. So in a fish, mercury can actually affect their reproductive rates, hormone concentrations, behavior, growth, and pathologies. One of the major effects that mercury has on the fish is that it causes an error in their brain development, which makes them fail to produce enough dopamine. Humans have a similar condition to this called Parkinson's, where a human brain doesn't produce enough dopamine, and it causes really severe symptoms. Fish who are having been exposed to mercury and have this dopamine lack actually experience similar symptoms to those a Parkinson's patient would. So to end this video, I would just like to thank Dr. Roberts one more time for talking to us interns. It was so exciting to learn about your research and really interesting to learn about your research methods. And then I would also like to thank everyone watching this video, and I hope to see you guys next time.